Welcome back, everyone. So we're going to finish off Chapter 7, Patterns of Inheritance. This will be also in Campbell Biology. This will cover Chapter 12. Okay, so we're switching chapters in Campbell Biology. So again, we're finishing off Chapter 7, Patterns of Inheritance, with 7.3 extensions of the laws of inheritance. So in this final section of Chapter 7, we're going to look at some of the exceptions to the laws of inheritance that we saw with Mendel's experiments. So remember how in Mendel's experiments we saw, for example, according to Mendel's law of independent assortment, genes sort independently of each other in gametes during meiosis. And that occurs because chromosomes on which the genes reside assort independently during meiosis. And crossing over causes most genes on the same chromosome to behave independently. But what happens when genes are located close to each other on the same chromosomes? They will, be inherited together. So we're going to see how, there's exceptions to those rules, those laws. And we're also going to see that, there's other things that violate, the experiments that Mendel, saw in his experiments. So we're going to, by the end of this video, you should be able to identify non-Mendelian inheritance patterns such as incomplete dominance, codominance, multiple alleles, and sex linkage from the results of crosses. You should also be able to explain the effect of linkage and recombination on gamete genotypes. And then finally, be able to explain the phenotypic outcomes of epistatic effects among genes. Okay, so this will be a long video just because we're covering quite a few topics, okay, with all these different non-Mendelian inheritance patterns. So just to kind of review what we learned from Mendel's experiments with his pea plants, we learned from those experiments that there are two types of units or alleles that exist with every gene, and that those alleles maintain their integrity in each generation. Okay, so there's no blending of the two. There's, a dominant and a recessive, and if both of them are present in the offspring, the dominant will dominate in the phenotype and the recessive will stay hidden with no contribution in the phenotype. And so, for example, in the pea colors, if we have a heterozygote, the pea color will show yellow due to the dominant allele masking the effects of the recessive allele. So the only way that we can show the recessive trait is if we have two recessive alleles, okay? So we can call the heterozygotes the carriers because they carry the recessive allele, but they show the dominant phenotype, okay? And so sometimes with the heterozygotes, since they carry the recessive allele, we can then see in their offspring, they might exhibit the recessive phenotype, okay? So these are, the patterns that we're seeing with Mendel's experiments. However, we don't always see that in every single everyday, gene with every single gene experience, okay? So we don't always see that there's only two types of alleles that exist for every gene. We don't always see that the dominant allele is always dominant to the recessive allele and that the recessive allele is always hidden. So we're going to see some exceptions to the rules, okay? So there are alternatives to dominance and recessives. So when we look at Mendel's experiments, his experiments are what we call simple patterns of dominance and recessive in a single gene model. And so we refer to his particular experiments as basically complete dominance. So that just means that the dominant allele is completely dominant over the recessive allele. And so in this case, like the yellow allele is completely dominant over the green allele. And so in the heterozygote, you will only see the yellow showing, right? So you only see yellow seed color. The green is being masked by the yellow. But the green is still there, right? That green allele is still there because you see it being passed to the offspring. And so if the offspring exhibit a homozygous recessive condition, then you see the green showing up in the offspring, okay? So we're going to see some exceptions to dominance and recessiveness. So there's several important modes of inheritance that don't follow that single gene model. So we're going to take a look at something called incomplete dominance, something called codominance, something called multiple alleles, and something called sex-linked traits. So we're going to start out with an Amoeba Sisters video. This Amoeba Sisters video will cover the first two, incomplete dominance and codominance, as well as the topic of polygenic traits and epistasis, which we'll cover later on in the topic. Okay, so we're going to kind of skip around. So we'll talk about these two as well as a topic later on. And then after the video, we'll go into detail about these two concepts. Okay? So let's get into it.
What do snapdragons, human height, and speckled chickens have in common? Well, they are easily identified as non-Mendelian traits. By non-Mendelian, we mean that genetically, they are rule breakers. They don't follow the regular Mendelian rule that having a dominant allele means the dominant trait will show. Remember in our guinea pig video, having a dominant allele represented by a capital H meant that the guinea pig would have hair? Only if there was no dominant present, a genotype of little h, would there be a hairless guinea pig. Well, that's a Mendelian trait. What we're going to cover now is when these traits are non-Mendelian and they don't follow these basic rules. So let's first start by snapdragons. We confess that when we heard this word, we thought they were some really amazing kind of creature. Well, they are amazing, but they're flowers, so I don't know, that's not exactly what we envisioned. Well, in snapdragon genetics, there can be three phenotypes: red, white, or something in between, pink. It's called incomplete dominance. In incomplete dominance, the dominant allele is not completely expressed when the recessive allele is around. There isn't really a clear dominant allele. If you cross a red flower, and we're going to write it here, big R, and a white flower, little r, you are going to get offspring that are big R, little r. But unlike a Mendelian trait, if this is incomplete dominance, that big R allele is not completely expressed when the little r is around. So big R, little r in this case is pink. If you cross two pink flowers, big R, little r, like shown in this Punnett square here, you can get offspring that are red, white, or pink. Incomplete dominance is different from codominance. Codominance, like a co-worker, that prefix co should make you think together. They work together, the alleles, that is. For that reason, we like to use different letters entirely to represent the alleles. There is a codominance involving color in some types of chickens. Take a look at this Punnett square. If you cross a black chicken, represented here by BB, and a white chicken, represented here by WW, all the offspring here are BW. BW chickens are both black and white. They're speckled. See, both traits show up. This is the essence of codominance. And you know, what's more awesome than a speckled chicken? Well, except for those silky bantam chickens. By the way, you will find that many people use different formatting for incomplete dominance and codominance. Some people like to use different letters entirely for incomplete dominance, and some people don't. Some people also prefer to do exponents with codominance, and some people don't. Formatting aside, when you're solving these Punnett squares, since we find this really does vary from classroom to classroom, the real concept is that in incomplete dominance, one allele is not completely dominant over the other. So you see this almost in-between phenotype. In codominance, both alleles are expressed. It's the application that matters here. Height is fascinating. In our immediate family, Pinky is taller than Petunia. Our mom is also taller than Petunia. So how does that happen? There isn't just one height gene. There's a lot of genes that determine your height. What I mean by that is that you don't just have a pair of alleles, like big A, that will code for your height. It's more like someone having a genotype of big A, big B, little b, big C, little c, and maybe more genes to ultimately determine your height. And you inherit one allele for each of the height genes from each parent. All of these genes work together to determine your height. Your skin color is also determined by many genes, just like your height. These are called polygenic traits. Poly means many, so many genes coding for one trait is what polygenic means. By the way, both height and skin color can be influenced by environmental factors as well. Nutrition, growing up, can affect your height, just as spending a lot of time in the sun can affect your skin color. However, this doesn't change the genetics for this trait. And finally, one more thing that we want to talk about. Epistasis. Epistasis is when one gene really depends on another gene for it to even be expressed. I mean, it really depends on this other gene. So let's pick an animal like, a llama. Let's say this llama has a dominant big B allele, which means its wool will be black. So big B, or big B, little b means that it will have black wool. And let's say that if a llama has a pair of recessive alleles, little b, then it will have brown wool. Now, what if there is another gene that controls whether this pigment will even be expressed in the llama in the first place? In this hypothetical example, a llama could have this other gene coded for by big C, big C, little c, or little c in its genotype. However, if a llama has the genotype little c, it will not allow the other gene for wool color to even be expressed. Since we have two genes here, the gene for wool color and then this gene that controls whether the color is even expressed, this calls for our favorite 16-square dihybrid cross. If you notice in this dihybrid, crossing two heterozygote llamas, any llama with big B and big B, little b will typically give a black llama, and little b will typically give a brown llama. In all cases, unless they inherit a little c. If the llama has a little c in its genotype, then the gene for wool color is not expressed, and the llama is albino. This means that no pigment is expressed at all. Pretty interesting. There are many other non-Mendelian traits. We have another video on multiple alleles and a video on sex-linked traits. These are also non-Mendelian. It's kind of fascinating to see what can happen when you have these rule breakers. One last thing. Keep in mind that when problem solving in genetics, you do not want to just assume it's non-Mendelian, unless you are provided information or clues in the problem that it might be. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so again, that was a great introduction into both incomplete dominance and codominance, as well as polygenic and epistasis, which we'll talk about later. So from the video, we learned about incomplete dominance. So what is incomplete dominance? So it's basically the heterozygote is showing kind of a blending of the two traits. So instead of the dominant allele being completely dominant, masking the recessive allele, the recessive allele is still kind of showing through, and it kind of blends them together. So the perfect example would be in snapdragons. So in snapdragons, if we cross a white flowered snapdragon with a red flower snapdragon, the offspring will actually be pink. Okay, so If we look at the genotypes, and again, from the video, they stated perfectly that, there's different ways to write the information down. Your book is going with superscripts, okay? So they have the characteristic labeled color, and then they use the superscript W for white and red for red, or R for red. You can go with the way that they did it in the video, where they just did R for red and little r for white. So it doesn't really matter how you do it. I actually, I'm one of those instructors that I don't care. I'm not strict when it comes to the way you write things out. So if you want to set up your Punnett square where you use letters without superscripts, if you just want to stick with uppercase and lowercase, or if you want to stick with different letters, that is fine, okay? As long as you understand what you're doing, that's perfect, okay? The only thing I really care about is that when you're done, you write things out, okay? So, for example, here we have a cross between a homozygous parent with white flowers and a homozygous parent with red flowers, okay? So in this case, the heterozygote would end up with an allele from the red flower plant and an allele from the white flower plant. And so in this case, because they have a red allele and a white allele, neither of these colors are going to be completely dominant over each other, so they blend together. And what happens when you blend red and white together? It produces the color pink. So the offspring's phenotype will be pink. Now, what happens when we cross a pink flower plant with a pink flower plant? Okay, so we can set up a Punnett square to kind of demonstrate that. And unfortunately, I didn't leave enough room to really do that. So I'm going to go ahead and just kind of do a Punnett square down here. Hopefully there'll be enough room. And again, you can do superscripts like this, or you could just do the R and the W like that. R, W. So I'm just taking two pink flower plants. So this is pink, and then we're going to see what happens when we cross the two, okay? So bring the alleles across and the alleles down. So I'm going to bring the alleles down, red, red, white, white. Bring this one across, red, red, white, white. So what do I end up with? I end up with one red plant. I end up with two pink. And one white, okay? So you can see by crossing the pink flower plants with each other, you end up with different offspring. You end up with about 25% of the offspring will be red, 25% will be white, and then 50% will be pink. Okay, so in incomplete dominance, the heterozygote has, one copy of the red allele, one copy of the white allele, okay? So, kind of interesting how we see the blending of the two traits to create kind of an intermediate phenotype in the heterozygote. All right. All right, so let's move on to codominance. So in codominance, this is different from incomplete dominance because both alleles are expressed simultaneously. So instead of blending the two together to create an intermediate phenotypes, you're going to see both of them expressed, okay? So remember the speckled chicken, okay? So in the speckled chicken, we don't see a gray chicken when we blended when we had the black chicken mated with the white chicken. Instead, we see a black and white polka dot chicken. Okay, so we see black spots and white spots on that chicken. Okay, so that's an example of codominance. So codominance means that we're expressing both alleles equally. Okay, so we're not blending them together to form an intermediate like we did with incomplete dominance. So that's different. Okay, so here's another example. We have blood types. Okay, so your blood type, there are different blood types. We're just giving two types here. There's actually three. There's actually four types of blood, but we're not getting into that just yet. But for homozygotes, we have two examples of homozygotes for a blood type. We have homozygotes for type A. So we have a superscript again given. So type A is I with a superscript A, and then type B, superscript B, superscript B. So somebody with type A that's homozygous for it has two A alleles. Type B has two B alleles. That's homozygous for it. Okay, and then they're expressing, if they're type A, that's homozygous, they have the A phenotype. If they're homozygous B, they have the B phenotype. Okay, and let's say that we cross, if we have a heterozygous for that's AB. So if you're type AB, that means you have an A allele and a B allele. You express both phenotypes equally. Okay, so you actually have, basically what blood typing means is that you have, if you're type A, you have a special A carbohydrate that's on the surface of your blood, of your red blood cell. If you're type B, you have the B carbohydrate on the surface of your red blood cell. Okay, if you're type AB, you have both carbohydrates attached to your red blood cell. If you're type O, you'd have neither. Okay, and so an individual that has AB, they have both types of the molecules attached to the red blood cell. They have the A type and the B type. Okay, they don't have a blend of the two. They have both types expressed. Okay, so codominance, we're expressing both types. They have the A carbohydrate and the B carbohydrate attached to the surface of the red blood cell. Whereas somebody with type A only has the A carbohydrate attached. Somebody with type B only has the B carbohydrate attached. Type AB has both. All right, so think codominance, both alleles are expressed in the phenotype. Incomplete dominance, you're blending the two together to form an intermediate phenotype. All right. So we've talked about incomplete dominance and codominance. So the next on our list, we're going to get into multiple alleles. So with multiple alleles, we're also going to talk about blood typing, because with blood typing, we have type A, we have type B, we have type AB, and we have type O. So we're going to start out with another Amoeba Sisters video that will talk about our multiple allele system with our blood typing.
There's a lot of phenotypes that are easy to tell. Your eye color, your hair texture, your height, whether you have a straight thumb or hitchhiker thumb. But one phenotype that you can't tell just by looking is your blood type. Your blood is really made up of many things: platelets, plasma, red blood cells. But you've probably heard before that when blood is donated, it's very important that it is matched correctly. And that's true because blood type phenotypes vary. It really boils down to the fact that red blood cells, they're not naked. They have proteins on their surface. And it turns out that your immune system is very protective, and if it gets blood that's donated that has different proteins that it's not used to, it will attack them. With blood type, you can have several different phenotypes: A, B, AB, or O. These letters stand for antigens that are found on the red blood cells. So type A blood, for example, has A antigens on the surface of red blood cells. Type B blood, for example, has B antigens on the surface of red blood cells. Type AB has both A and B antigens on the surface of red blood cells. Type O, I like to think of it looking like a 0. It doesn't have A or B antigens. It's naked. Well, okay, it does have other proteins on its surface, but not A or B. So think of O as looking like a 0. Doesn't have A or B. So if you have type B blood, you have B antigens on the surface of red blood cells. That means a person with type B can accept another person's type B blood because B is an antigen that their body recognizes. But if you try to give that person a type A blood type, that's an antigen that the immune system does not recognize. That person's immune system would attack. It would also attack AB blood because that includes the A that it doesn't recognize. Now type O would be safe. Remember, O, think of like a 0. It doesn't have A or B antigens, so type O can donate to everyone. Now, while O individuals can donate to everyone, they can only receive blood from another type O. Because type O doesn't have A or B antigens, so their immune system will attack any other blood type that does. Neither of us have type AB blood, but we would think this is a cool blood type to have in the sense that you could receive blood from anyone. If an AB person received blood from a person that had type A, well, they've got the A, so it's all good. And if an AB person received blood from a person that had type B, well, they've got the B antigen too, so it's all good. They can receive blood from type O too because there's not any antigens to even worry about. Now, one thing we do want to add that makes all of this a bit more complicated. Blood types also have a plus or minus sign listed by the blood type. This makes a big difference with blood donations. If you have a plus, it means that you have this other little protein called Rh factor on the surface of your blood cells. And if you have a negative, it means that you do not have this little protein called Rh factor on the surface of your red blood cells. Now, we're not going to be able to go into that in this short clip, so to the Google for that if you would like to learn more about this. Blood type is genetically inherited, and a great example of multiple alleles. Remember that alleles are a form of a gene, like a flavor. Just an analogy. So let's put this into practice. Let's say a couple gives birth to a baby boy. Both parents have type A blood, but then there was a mix-up at the hospital. And now, sorry, I get really into my drama. There are these two baby boys, and the hospital does not know which one belongs to the couple. Probably not a hospital where you want to have kids, but let's try and use our blood type genetic problem-solving skills to help us out here. Baby Phil has type B blood, and baby Sylvester has type O blood. Could either of these babies belong to the couple who both have type A blood? Well, let's talk about phenotypes and genotypes. The phenotype of type A blood is A, but the genotype is written like this or this. The format of writing here helps with multiple alleles like blood type problems, and we can show you why when we work this out in the Punnett square. Now, you may notice that I said that blood type A could be written this way or this way. Without testing, we don't really know which one it is. You can consider this one to be homozygous and this one to be heterozygous. The phenotype of type B blood is B, but the genotype is written as this or this. The phenotype of type AB blood is AB, and the genotype is written this way. There's no other way to write that one. The phenotype for type O blood is O, and the genotype is written like this. Remember how I said that the O kind of looks like a 0, and so you can think of it as having 0 blood type antigens? Well, that's what it is. Little I, no coefficient. Okay, back to the babies. We are told the parents both have type A, but remember, we don't know whether that means they are this type A, the homozygous, or this type A, the heterozygous. And the mom could be one of those genotypes, and the father could be the other genotype, or they could both be the same genotype. We just don't know. Okay, so let's try it out with all of the possibilities. Remember that we write the parent genotypes on the top and the side of the Punnett squares, like this. We'll fill in the Punnett squares and we'll make sure to have capital I's first, just for formatting purposes. Okay, so is it possible to get baby Phil, who has type B, in any of these offspring? Nope, he must be someone else's baby. What about baby Sylvester with his type O? Yes, it is possible, but both parents would have to be heterozygous A. That means they both must be this. Then yes, you would have a 25% chance, that's 1 in 4 here, of having a baby with type O blood. Of course, if I was advising this couple, I would also insist on the DNA test, as this blood type problem only shows that it's possible that baby Sylvester is theirs. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so from that video we learned not only about multiple alleles, but also how our blood typing system includes codominance and just the regular old dominant over recessiveness. So we're going to get into all of that. Just a reminder, when we're talking about these populations with the genotypes and the phenotypes with multiple alleles, occasionally you'll see the most commonly occurring genotype or phenotype is labeled as a positive, and that's known as the wild type. So it's just labeled in your book like this. We don't actually get into this until we talk about fruit flies, but just be aware that it's labeled in your textbook under multiple alleles, so that's why I'm just going to bring it up now. So a wild type is the most common. For our blood types, we don't actually have a most common. I guess type O is about 40% of the population, but still that's 40%. The others are pretty evenly distributed. I think AB is the least common, but that's still fairly, we're pretty evenly spread. So again, with our blood typing, we have 4 different, let's start out with 3 different alleles present. We have the A allele. So the A allele codes for that A antigen or molecule on the surface of our red blood cell. The B allele codes for the B antigen or molecule on our red blood cell. And then the I allele codes for no antigen or molecule on our red blood cell. And then, as the video talked about, the A allele and the B allele are codominant to each other and are both dominant over the I allele, okay? And so that's why we have an uppercase I with the superscript A, okay, for the A allele, and we have an uppercase I with a superscript B for the B allele, and then we have a lowercase I for the, what we think of as the O, okay? So just, that's why we have those. Some students prefer to just do the A's and the B's and the O's. However, just if you are going to go that route, make sure you remember that O is recessive to A and B, okay? So just remember that, okay? So when we do set up Punnett squares, we do need to make sure that we're including the superscripts and the little i's and the big i's when we do set them up. So if we were to do an example of a Punnett square, let's say that we have a again, like what she talked about with the video, if you're type A, your phenotype is A, you could possibly be heterozygous, so that means that you have an A allele and an O allele, or you could be homozygous recessive, you have 2 A alleles, or excuse me, homozygous dominant, two A alleles. If you're type B, you could be heterozygous, or you could be homozygous for the B. If you're type AB, we know your genotype. You have an A allele and a B allele. And if you're type O, we know your genotype. You have two recessive alleles. Okay? And then she talked about like donation and you know, blood types and stuff like that. So if you're type O, you're considered the universal donor because of the fact that you don't have any of those antigens on your red blood cells. So you can donate to type AB, type B, or type A blood type. If you're type AB, you're the universal receiver. You can receive anybody's blood because you have all the antigens. So you can receive type A blood. You can, because you have the A antigen. You can receive type B blood because you have the B antigen, and you have type O blood because they don't have an antigen. You can receive that. If you're type A, you cannot receive B blood because your antigen would react to that and cause clotting. You cannot receive AB blood because the B in that would cause clotting. Okay. If you're type B, you cannot receive A or AB blood. All right. So that's a little bit about that. She also mentioned the Rh factor. So the Rh factor is just another addition in your blood. So you're either positive for that or negative for that. Okay. And that one's also like a dominance or recessiveness. So if you are positive for it, you can receive, it's the same thing, like you can receive anybody's, like if you're A positive, you can receive, you know, O positive or O negative, A positive, A negative blood. So you can receive both positive and negative Rh factor blood. If you're Rh negative, you can only receive negative blood. Okay, the positive will react with you. All right. So that's just some interesting, like, you know, medical stuff if you're interested in stuff like that. All right. And I believe in your lab kit, you actually do get one of those antigen testing kits. So you do get to test to see what type of blood you are. So you can check and see if you are type A, B, AB, or O. All right. So now that we've, you know, talked about codominance and multiple alleles, we're going to move on to sex-linked traits. So with sex-linked traits, we're going to be talking about the sex chromosomes. So far we've only been sticking with the autosomes, chromosomes 1 through 22, but what about those sex chromosomes, the X's and the Y's? So we're going to start out with an Amoeba Sisters video that will give us an introduction into those.
Would you like to know one of the most underappreciated pieces of cytoplasm out there? Platelets. We take for granted the function of our platelets, which are fragments of cells that help stop us from bleeding. They help our blood to clot when we get hurt. But there is a disorder called hemophilia that can affect those platelets, and therefore the ability of blood to clot. With hemophilia, even a small cut could be dangerous because the bleeding can be continuous. There are now many good treatments for the symptoms of hemophilia that have greatly improved outcomes with this disorder, although it wasn't always that way. Hemophilia is a sex-linked recessive trait, which means it is different from general Mendelian genetic problems. With sex-linked traits, we still use the terms dominant and recessive for alleles, except this time, those alleles are on sex chromosomes. This is the case with sex-linked traits. What is a sex chromosome? Well, first take a look at this karyotype, which shows a human's chromosomes. This karyotype has 46 chromosomes arranged here in 23 pairs. 46 is the general number of chromosomes in humans, but as we mentioned in our karyotype video, individuals can have more or fewer than 46. Chromosomes are made up of DNA and protein. They contain your genes. 23 of this person's chromosomes came from an egg cell, and 23 of this person's chromosomes came from a sperm cell. The last 2 of the 46 chromosomes in this karyotype are called sex chromosomes. The sex chromosomes are commonly called X or Y chromosomes, but X and Y have nothing to do with the shape of the chromosome. The reason for the X and Y names is really interesting. Check out our further reading to learn more. All humans have at least one X chromosome. In most cases, females have a combination of XX and males have a combination of XY, but individuals can have additional or fewer sex chromosomes. We're focusing on sex chromosomes in this video because we're going to show how to do Punnett squares with traits on these chromosomes. In the case of Punnett squares with sex-linked traits, the traits being tracked tend to be on the X chromosome and not the Y chromosome. The X chromosome is much larger than the Y chromosome and contains more genes than the Y chromosome, so technically this video will be about X-linked traits, but we do want you to know there are Y-linked traits too. Referring back to the beginning, when we mentioned the disorder hemophilia, hemophilia is a sex-linked recessive disorder carried on the X chromosome. So focusing on that, let's learn how to solve a Punnett square that involves this trait. We will use the capital letter H to represent an allele for not having hemophilia, and a lowercase letter h to represent an allele for having hemophilia. Why? We're going to do that because we mentioned hemophilia is a recessive sex-linked disorder, which is why it's being represented by a lowercase letter h. Only it must be placed on the X chromosome as a superscript, like an exponent. Let me explain what I mean by that. Let's consider a female individual shown with XX here. If the genotype is X big H, or X big H, X little h, this female will not have hemophilia. Why? Because as long as there is at least one dominant allele, that dominating allele will be what shows in this trait. So, no hemophilia. Hemophilia is a sex-linked recessive disorder. However, the X big H, X little h genotype would be a carrier, meaning while the person wouldn't have hemophilia, they would be carrying the recessive allele. The only way for this particular female to have hemophilia would be the genotype X little h, because only when there is no dominant present will that recessive show up, at least in this type of trait. Let's consider a male individual, shown with XY here. X big HY would be a genotype of a male without hemophilia. Notice how I didn't put anything on the Y chromosome. Again, most sex-linked traits are on the X chromosome. If this male had the genotype X little hy, then this individual would have hemophilia. Since there aren't two X chromosomes for this person, either this person has hemophilia or doesn't. Let's try a Punnett square problem with this trait. Let's say these two people here both do not have hemophilia, although the female is a carrier of hemophilia. If they together have a biological child, what is the percent chance of the child having hemophilia? Also give genotype and phenotype ratios. Step 1, always determine the genotypes of the parents first. The female must be X big H, X little h. Why? The Punnett square problem says the individual doesn't have hemophilia, but is a carrier. That means this heterozygous genotype. It says the male does not have hemophilia, and so the genotype must be X big HY. If it was lowercase h, this male would have hemophilia. Step 2, place one parent's genotype on the top, outside the square like this. Place the other parent's genotype on the left, outside of the square like this. Step 3, fill in the square. For formatting purposes, place X chromosomes before Y. You also write any sex chromosomes with dominant letters first. The results you get in the squares would be the offspring, the babies. The genotype ratio could be written out like this. And the phenotype ratio, remember that these are the traits, can be written out like this. There's a 75% chance that a child will be born without hemophilia, and a 25% chance that a child will be born with hemophilia. Remember, like all Punnett squares, these are representing probabilities. So because it's a probability and not necessarily the exact outcome, they could, for example, have quadruplets that all do not have hemophilia, or they could have quadruplets that all have hemophilia. The probability of the latter is less likely, but it's still possible. 5 things to keep in mind when you are working on these kind of Punnett squares. Number 1, you do not want to just assume a trait is a sex-linked trait, because many traits are not sex-linked. More traits are found on the autosomes, actually. Autosomes are all the chromosomes that are not sex chromosomes. When you first started practicing with Punnett squares, it's likely those were problems that were not sex-linked, and thus the sex chromosomes of the parents would be irrelevant on the square. So never assume the problem should indicate it's a sex-linked trait in some way. Number 2, did you notice how the child in our example that would have hemophilia was XY? Sex-linked recessive traits are more common in XY genotypes compared to XX because XY has only one X chromosome. For example, some forms of color blindness are sex-linked recessive. It is more common in males. When arranging a pedigree that is tracking a sex-linked recessive trait, it can be common to see a lot of shaded squares in the pedigree, representing males that have the sex-linked recessive trait. You can learn more about pedigrees in our pedigree video. Number 3, you may be wondering, our example was a sex-linked recessive trait. But can there be a sex-linked dominant trait? Yes, a sex-linked dominant trait means it would only take one dominant allele for the individual to have the trait. Let's use the letter D to illustrate a hypothetical sex-linked dominant trait. If it was a sex-linked dominant trait, these two female genotypes would have the trait, because again, it only takes one capital letter, a dominant allele, to have this dominant trait. This female would not have the sex-linked dominant trait. This male would have the dominant sex-linked trait, and this male would not. Number 4, because we talked about the inheritance of the disorder hemophilia, and we also mentioned color blindness, we do want to make sure you don't leave with a misconception. Not all disorders that have a genetic component follow a one gene kind of trait. In fact, many don't. An example? We both talk about how we developed preeclampsia, a disorder that occurs during pregnancy or the postpartum period that can be life-threatening to those that develop it. While current research is studying genes that could play a role in developing preeclampsia, like genes that impact the placenta or genes that involve the vascular endothelium, the cause of preeclampsia is overall still not well understood. Many disorders, though, that have a genetic component can have multiple genes interacting together and wouldn't be something that you could place in a general Punnett square. And these disorders can have external factors as well that are separate from genetics. Number 5, our example was for humans. Humans are animals, but many animals do not have X and Y sex chromosomes. For example, birds, where it's Z and W, or some animals might have X and Y sex chromosomes, but maybe they typically have 10 of them, like the platypus. More in our description. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
Okay, so again, that was a good introduction into our sex-linked traits. So as a reminder, we as humans have 23 pairs of chromosomes. And so far, we've been focused in on our 22 pairs of homologous autosomes. So we have not been talking about the sex chromosomes at all. So all of the genetics, Punnett square problems have been assuming that they're autosomes, and therefore we haven't been, thinking about them, being XX or XY. So when we're talking about our sex chromosomes, we can have traits that are found only on the X chromosome. And since males only have one copy of the X chromosome, this can be an issue in the way that things get inherited. Okay, so just a reminder, females are homologous for the sex chromosome because we have two copies of it, XX. Males, you're non-homologous, so you only have one X chromosome. And as we saw from the video, the X chromosome is a lot bigger than the Y chromosome. So the X chromosome carries thousands of genes. And, not all those genes have to do with the gender, okay? It has other things involved, such as the hemophilia gene, okay? Or whether or not you're going to have hemophilia. And so, females have two copies of the, whether or not you're going to have hemophilia. Males only have one copy. So if that particular copy on males say that they're going to have hemophilia, they're going to have hemophilia. For females, they have to inherit two of those genes that say hemophilia in order for them to have hemophilia. So that's why males are more likely to be affected by sex-linked traits than females, okay? In your textbook, they give the example of fruit flies, and so they bring back this wild type example. So remember, wild type just means that it's the more common. And so in fruit flies, the more common eye color is red, Okay, so here's our two types of fruit flies. So red eye color is more common than white eye color, and it happens to be found on the X chromosome. And so females have two X chromosomes, males only have one X chromosome. And so when we're dealing with the sex-linked traits, we do write out the sex chromosomes, the X's and the Y's, and then we denote the particular gene that we're talking about as a superscript. And in this case, it's just a normal complete dominance issue. So we have the red allele is completely dominant over the white allele. So we do a big W for red and a little w for white. Okay, so we're doing that as a superscript. And we have to include the X's and the Y's. Okay, so this is not an option this time. Before I said, you could leave out the superscripts if you wanted to. This one, it's not an option. Okay, you have to include the X's and the Y's because for males, you don't have, you have two separate chromosomes, so we have to indicate that. Okay, so again, in this particular example, we have the male fruit fly is considered hemizygous. So hemizygous means only one allele because they only have one X chromosome. So in this case, they either have one copy of the red allele or one copy of the white allele. So their Y chromosome does not contain that gene because, the Y chromosome doesn't have that gene at all. So we leave the Y chromosome alone. It doesn't possess that particular gene at all. All right? Females have two copies of the allele because they're homologous. And so they could be homozygous dominant, so big W, big W. They could be heterozygous, big W, little w. So these two would have red eyes. Or they could be homozygous recessive, little w, have white eyes. Okay? So for males, they cannot be carriers. They cannot be heterozygous. They either have red eyes or white eyes. There's only two options there. Females, they could be, there's three options there. All right. So we're going to watch a video that is from your textbook on how to set up the sex-linked traits, Punnett squares, and then we'll set one up with as an example.
Welcome back to Biology. This week we're going to discuss the concept of sex-linked traits, specifically hemophilia. To begin with, remember that every somatic cell in the human body should contain 46 chromosomes or 23 pairs. We get one set of genes from our mother and one set of genes from our father. When paired together, the corresponding chromosomes from each parent are called a homologous pair. One of these 23 chromosome pairs determines the biological sex of the organism. The larger, longer type is called an X chromosome, and the smaller, shorter one is called a Y chromosome. Females have two X chromosomes, and males have one X and one Y. In humans, the female's eggs always contain an X chromosome, but the male's sperm may pass on either an X or a Y. The male determines the sex of the offspring. If he passes on an X chromosome, the resulting offspring will be female. If he passes on a Y, the resulting offspring will be male. The X and Y chromosomes both contain genes that help code for certain traits. Traits that are carried on either of these chromosomes are called sex-linked traits. For instance, the SRY gene on the Y chromosome codes for the development of the testes in males, and the AR gene on the X chromosome codes for male pattern baldness. Hemophilia is a classic example of a recessive X-linked trait. In a healthy individual, the body combats the hemorrhaging process first by constricting vessels at the site of injury. Then a complex series of events called the coagulation cascade activates structures known as platelets. Usually the platelets interlock to form a plug called a clot, which slows or stops the bleeding. In individuals with hemophilia, however, the coagulation process is disrupted. The platelets either do not mesh tightly enough, or sometimes they will not interlock at all. Untreated, this condition could cause potentially fatal bleeding, even from a very small wound. Individuals with hemophilia are almost exclusively male. So how is it possible that the genes for this condition are carried on the X chromosome rather than the Y? Hemophilia is recessive. Because females have two X chromosomes, receiving one with the mutation would not cause them to develop hemophilia. Their second X chromosome, carrying the dominant healthy gene, would overpower the recessive hemophilia gene. To develop hemophilia, they would need to receive the mutation from both parents, which is highly unlikely, about a 1 in 25 million chance. Men only have one X chromosome, and their Y chromosome can't overshadow the hemophilia gene. If their X chromosome contains the mutation, they will have hemophilia. Despite not actually suffering from the debilitating effects of hemophilia, women with one copy of the gene are carriers for the condition. They are capable of passing it on to their descendants. If they pass the hemophilia gene on to a daughter, the daughter would also become a carrier. If they pass the gene on to a son, he would develop hemophilia. If a carrier woman and a healthy man reproduce, they have a 50% chance of having a daughter who is a carrier for hemophilia, and a 50% chance of having a son who has the actual condition. One of the most famous examples of this happening is in the royal houses of Europe about a century ago. Queen Victoria of England was a carrier for hemophilia. She passed the mutation on to several of her children, and thus on to the royal houses of Germany, Spain, and ultimately Russia. Her granddaughter Alexandra married Tsar Nicholas II of Russia, and their son's hemophilia played a direct role in the downfall of the last Tsarist regime. Okay, that's pretty much it for this week. If you found this video useful,
Okay, so from that video, we saw basically a Punnett square set up as well as what's called a pedigree, and you'll see more of the pedigrees in the lab this week. So we'll talk a little bit more about how to set up a pedigree later on. But let's go ahead and take a look at an example of how to set up a Punnett square. So in this case, we're setting up a Punnett square with the fruit flies. And here we have a male fruit fly. In this case, the fruit fly has a dominant allele for red eyes, okay? And that one was crossed with a female fruit fly that is homozygous recessive for the white, okay? So we're going to simply put the female gametes on top, okay? So here's, the white allele here. There's the white allele there. And then the male's gametes on the side. So there's the red allele here, and then the Y chromosome there. And notice again, we're including the X's and the Y's, just because the Y allele doesn't have the Y chromosome doesn't have the gene, so we have to include the X's and the Y's as a place marker. Okay, we can't just include the W's without the X's and Y's, because then what do you do with the male that doesn't have that? It would be blank. And then you can see here how we bring the female's gametes down. So there's the little W, little W, little W, little W, little W, and then we bring the big W across, big W, big W, and then the Y across, Y, Y. So you can see from the Punnett square that there's a 50% chance that we have female, 50% chance that we have a male. And then we can see the genotypes. We have 50% chance that we have a carrier, and then 50% chance that we have basically a male with white eyes. So 50% chance that red-eyed carrier, 50% chance that we have a male with white eyes. Okay? So that's how we're going to set up the Punnett squares. So we have to include the sex chromosomes, and then we're going to include the alleles as superscripts on top of just the X chromosome. So let's do this visual connection question together. So what ratio of offspring would result from a cross between a white-eyed male and a female that is heterozygous for the red eye color? So first we have to write out the genotype. So a white-eyed male. So the male is XY. So what would I put as the superscript here? So if it's white-eyed, it's going to be lowercase w. Okay, and then we have a female here. Remember, I'm not putting anything on the Y chromosome. So for the female, it's heterozygous for red. So XX. So I'm going to do uppercase W and then lowercase w for red. Okay, so now I'm going to set up my Punnett square. So I'm going to put Dad over here. So he has lowercase w, y, and then Mom over here, uppercase w, and then lowercase w. Okay, and then I bring Dad's gametes across. X, lowercase, y, Mom's gametes down, X, big W, X, big W, Mom's gametes down, X, little w, X, little w. All right, so we look at the females first. Okay, so here we have a heterozygote. Okay, so we have one female that's heterozygous. Oops, I should do a big W, little w. So she has red eyes. I have one female that is white-eyed. I have one male that's red-eyed. Maybe I should write the phenotypes. This is female, female, male. And then I have one male that's white-eyed. So basically I have one of each, right? And this one's a carrier, by the way. She's heterozygous. Very bad. Bad writing, sorry about that. Okay. So what's the ratio of offspring results? So basically it's kind of hard to do a ratio when you're when they're all different and you have gender in here. But basically we have one carrier female with red eyes to one white-eyed female to one red-eyed male to one white-eyed male. Okay, and then remember the males are hemizygous, meaning that they just have one allele, so they're either red or white. There's no carriers involved there. Okay, so that's how we solve the sex-linked trait questions. So you have to include the X's and the Y's. Okay, if we didn't include the X's and the Y's, we would get a completely different result. All right, so moving on. So now we're going to switch into some other things that don't really have to do with the like Punnett squares necessarily. We're going to talk about linked genes that violate the laws of Mendel's law of independent assortment. So if we recall earlier, we talked about Mendel's law of independent assortment. So that states that genes, they don't influence each other with regard to sorting of alleles into gametes. So remember during middle metaphase part 1, the homologous chromosomes, they align themselves independently of each other so that every possible combination of alleles is equally likely to occur. So this applies to genes that are located on separate non-homologous chromosomes. But what happens when we have genes that are located on the same chromosome? So they can possibly be linked together and inherited together. So that's something called linkage. It's a phenomenon in which alleles that are located in close proximity to each other on the same chromosome are more likely to be inherited together. So an example of a linked gene would be if you think of people with red hair color and freckles. So those two genes happen to be like the freckle gene and the hair color gene are actually fairly close together, and so they tend to be inherited together. So that's why you see people that have red hair tend to actually have a lot of freckles. And it's just because most likely the two genes are going to get inherited together because they're located on the same chromosome in close proximity to each other. Now, it's not always inherited together because we do have something called the crossing over effect. But if the genes are located close together, there's a higher likelihood that they will be inherited together, okay, due to this linkage effect. All right? So if we recall from meiosis, when we talked about prophase part 1, there's a phenomenon called the crossing over effect. So remember how in crossing over, the homologous chromosomes get so close together that one homologous chromosome could potentially flop over the other one and break off and attach to the other one, okay? And so we end up with what's called a recombinant chromosome. So recombination is the process during meiosis in which homologous chromosomes exchange linear segments of genetic material, therefore dramatically increasing genetic variation within the offspring and separating linked genes. Okay, so the closer the genes are together on the chromosome, the more likely they're going to get inherited together. So for example, in this piece here, chromosome A and B are more likely to get inherited together than say A and C. Okay, so A and C, they're further apart, so they have a less likely chance that they're going to get inherited together than A and B. So you can see how in this case, A and B, they got linked together, but then A and C, they are now on separate chromosomes. All right? So it just has to do with location of chromosomes within, or which will increase their likelihood of getting inherited together or not get inherited. So the further they are from each other increases the likelihood that they will not be inherited together. The closer they are, more likely they will be linked. All right? So linked genes, they can potentially be inherited together, which would violate the law of independent assortment. All right? So that's what that's all about. All right, and then finally we get into epistasis. So epistasis, the first video in our Amoeba Sisters video talk at the beginning of this video touched on epistasis. So epistasis is an interaction between genes such that one gene masks or interferes with the expression of another. So in that Amoeba Sisters video, she talked about the llamas, where we had the black color llama and the brown color llama are controlled by a gene, but then there's another gene that could either that could potentially turn the color off completely and cause albinism. Well, we have the same phenomena occur in mice. Okay, so in mice we have the wild type color, coat color, which is agouti, which is dominant to solid color fur color, which is recessive. So the agouti color, which is this brown color, is dominant to this black color. So that's one gene, okay? And then we have another gene which will cause the expression of the pigment to occur. That's dominant over albinism, which will be the lack of expression of the gene. Okay, so we have two things happening. We have the coat color, the agouti or black, and then we have whether the coat color will be expressed or not. Okay, so two different genes happening here. So let's say that we have these mice that are heterozygous for both. Okay, so they are agouti color. So they are heterozygous for coat color. So they have a dominant allele that shows the agouti color, but they carry the recessive black color. Okay, they also have the pigmentation allele that allows them to express the coloration, but they carry the albino trait. Okay, so here's their possibilities. Remember, we did that foil. So foil, foil, F-O-I-L. So if we take A, big A, little a, big C, little c, we do first, that's one possible gamete combination, outer, inner, and last. Okay, so those are the 4 possible gametes. Okay, so that's where the 4 possible gametes went. And then here's all of the different combinations. So notice that some of the combinations we ended up with the agouti coloration being expressed. We have the black coloration being expressed. And then here you can see that we should have the agouti coloration. The genes show agouti coloration, but because it's homozygous recessive for the whether it's going to be expressed or not, we show an albinism. Okay, so this is epistasis, where we have one gene masks the effect or interferes with the expression of the other. Because this is homozygous recessive, it doesn't matter what the first gene says. This gene says that no pigment is going to be expressed. Okay, so even though it's homozygous dominant, it should be agouti color. No pigment is going to be shown, and the mouse is going to be albino. Okay, same with down here. We should have a black pigmented mouse, but because we are homozygous recessive for pigmentation, we get no pigment being expressed. Okay, so this is epistasis, where one gene masks the effect of another. All right, so that was the result of that. Okay, so let's do some review questions. If black and white true breeding mice are mated and the result is all gray offspring, what inheritance pattern would this be indicative of? Is it A, dominance, B, codominance, C, multiple alleles, or D, incomplete dominance? So what inheritance pattern would that be if we ended up with gray offspring when the parents were black and white? If you said incomplete dominance, I would agree with you. So incomplete is the blending of the 2 parents. So black and white blended together equals gray. Here's another question. The A, B, O blood groups in humans are expressed as the A, B, and I alleles, or A, B, and O alleles. The A allele encodes the A blood group antigen, B encodes B, and I encodes O. Both A and B are dominant to O. If a heterozygous blood type A parent, so their genotype is AO, and a heterozygous blood type B parent, BO, mate, 1/4 of their offspring are expected to have the A blood type, so AB, in which both antigens are expressed equally. Therefore, AB blood type groups are an example of, is it A, multiple alleles and incomplete dominance, B, codominance and incomplete dominance, C, incomplete dominance only, or D, multiple alleles and codominance? If you said multiple alleles and codominance, I would agree with you. So we have three alleles here. We have the A allele, the B allele, and the O allele. And then we have codominance, where A and B are equally dominant to each other. Okay. So D is the correct answer. In a cross between a homozygous red-eyed male fruit fly and a white-eyed male fruit fly, what is the expected outcome? Okay, so normally in this type of situation, if you came across something like this on an exam, we would tell you which is dominant to which. Okay, so let's just go back. So remember in a previous slide it told you that, where did it say it? That the red is X big W is dominant over white little w, and then remember that this is a sex-linked trait. Okay, so we would give you that information, by the way. Okay, so just be aware of that. You're not expected to remember all of the questions, which ones are sex-linked, which ones are incomplete dominant, so we would have to give you some of this information. So in a cross between a homozygous red-eyed female, okay, so we know that she is homozygous red-eyed, okay, so big W, and a white-eyed male, what is the expected outcome? Okay, so you can set up a Punnett square. Sometimes you don't even need to do a Punnett square. If you get good at this, you kind of already can visualize what the outcome would be. Like, I kind of already know the answer. Okay. Just by kind of visualizing, especially since the female is red-eyed, homozygous. All right, so there are dominant alleles in all of the offspring because of mom, okay? So that means, they're all red-eyed, okay? All right, so we are done with chapter 7, patterns of inheritance. So for this section, 7.3, extensions of the law of inheritance, you should be able to identify non-Mendelian inheritance patterns such as incomplete dominance, codominance, multiple alleles, and sex linkage from the results of crosses. You should also be able to explain the effects of linkage and recombination on gamete genotypes. And then finally explain the phenotypic outcomes of epistatic effects among genes. So in the next video, we're going to get into lab topic 7, which deals with Mendelian genetics. So there it's basically Punnett square palooza. So we're going to be dealing with all sorts of Punnett square problems. We'll learn about how to set up all of these Punnett squares, from complete dominance to incomplete dominance to codominance to multiple alleles to sex linkage, and all of that, including pedigrees. So if you're still a little wary about how to set up a Punnett square, we'll get lots of practice this week in lab. Okay, so I'll see you there.